Hello, thanks for the invitation to speak about clinical trial preparation. Here are my disclosures. So the objectives of this session are to cover some practical steps for identifying and formulating a clinical trial question, for designing a trial, and for obtaining funding for a trial. And my brief was to talk from the perspective of junior investigators without substantial experience in this area before, but some of the comments, of course, will also apply for senior investigators. So first of all, how do we identify and formulate a clinical trial question? This is the critical thing, because without a good trial question, then you can put a substantial amount of effort in to no avail. So important to get right. So here are some tips on an initial approach to identifying a clinical trial question. So first of all, one thing you can do is look through recent trials in high impact journals, such as New England Journal of Medicine or the Lancet family of journals, Lancet HIV, to see if there's a gap in the evidence that might justify an additional trial that you might be able to do in your local setting. And differences in healthcare delivery and monitoring systems are generally better justification for doing a trial than simply repeating a trial in a different ethnic or racial group, unless there's a known genetic association with different responses to a drug that might justify that. And the other thing you can do to get some ideas is to check international treatment guidelines to see if they contain recommendations for high priority research questions that you might be able to address with your trial. So here's an example of this kind of approach, and it's about the Dawning trial. And let me explain to you about this. So here's the Dawning trial design. It was a second-line therapy trial looking at the best options for people who were failing on 2 nucleosides and efavirenz, and they randomized people to dolutegravir and 2 nucleosides or lopinavir ritonavir and 2 nucleosides and followed up for 48 weeks. That was the trial design, and this is what the paper showed in Lancet ID. It showed that dolutegravir was a clear winner over and above lopinavir and ritonavir. And it was so good that they stopped the trial after 24 weeks interim review. So all well and good, paper published, trial done, but is that the end of the story? No, if you look carefully at the trial design, you can find a number of things which actually impair the generalizability to many resource-limited settings. So for example, their entry criteria were people who'd failed with a viral load above 400 copies on 2 occasions, 7 days apart, whereas in most settings, you wouldn't switch someone to second-line therapy after just 7 days. And also at that threshold, you would usually do a period of adherence counseling and then check again. And in WHO recommendations, use a threshold of 1,000 copies. The other key thing with this trial is that they mandated resistance testing, again, not part of usual practice at first-line failure, and selection of 1 active nucleoside. And then the primary outcome was a viral load less than 50 copies. So a number of things that are a little bit different from clinical practice in many settings, certainly different from the public health approach. And again, on the same topic, to get ideas, you could check international treatment guidelines. So here is the last main WHO guidelines in December 2018. And you can see after each chapter of recommendations, there's a chapter or a subsection on research gaps. And so these can be very good sources of ideas for doing clinical trials. And just to look at the second-line research gaps here, and you can see here they're recommending that we need further evidence to inform the use of tenofovir 3-TC dolutegravir in second line after people fail on a dolutegravir-based regimen. And they're also recommending that we need more information on the impact of resistance on a nucleoside backbone in people going on to dolutegravir second line. So again, aligned with the conclusions one might have drawn from the Dawning trial. And this is a practical example. These considerations led to the design of another second-line therapy trial called Nadia, which addressed these nucleoside questions in a way that was relevant to resource-limited settings with different thresholds and so on. And this has been recently published. So again, these 2 approaches, looking at treatment guidelines, looking at published papers and looking for weaknesses, led to the design of a trial, a relatively successful trial. What else can you do to pick up a trial question? Well, you could approach your local pharmaceutical company team and ask them, well, okay, you've got this new drug or this licensed drug. how about this idea? Or what trials do you want to see done in my local population? They might have some thoughts about what they're really looking to get in terms of data. Or you could discuss common clinical dilemmas and think about those and discuss with your colleagues where you think the evidence is lacking in your situation, or what advantages do you have in your local situation? Maybe you have a higher proportion of certain risk populations that tend to be underrepresented in clinical trials, or this is always a good rate, high rates of co-infections that pose treatment challenges, particularly something like TB, where you've got all of the interactions with rifampicin. And it's important to think broadly, not just drugs. You may get good ideas thinking more broadly than that. So you can test in trials, you can test management strategies or prevention measures, all sorts of things you can test and think about for trial ideas. So that's about generating good ideas. What about evaluating these ideas? So here's my key recommendation is don't fixate initially on one idea. It's better to develop a number of possible ideas because this makes it easier for you to let go of a less good idea as long as you have backups. So for each of your ideas, write down, specifying for each what's the gap in the evidence and why the trial is really needed. What exactly is the trial question? What's the intervention and control? Is it a superiority trial or a non-inferiority trial? And then take these ideas and discuss with colleagues and experienced trialists to evaluate whether any or all of these ideas are valuable or how you might tweak them to make them a bit better. And then when you've got that, then select the best one to move forward with. So don't put all, as we say, put all of your eggs in one basket too early on. It's better to be open-minded, and then you might come up with an idea that's really very valid and strong. So what are the good attributes of a good trial idea? So it needs to be original. That's important. There's nothing worse than doing a trial, spending a lot of time and effort, and then you find 3 months before you complete your trial that somebody publishes exactly the same trial, and then there's little point in you having done your trial. So it needs to be original, it needs to be clinically relevant, preferably likely to impact practice. The trial needs to be durable, or at least the question needs to be durable. So it needs to be relevant a few years down the line, not just for the next few months. And it needs to be feasible in terms of recruitment and finances. So how about assessing whether it's sufficiently original? This is absolutely key before progressing further, as I've just explained. And what you must do is check trial registration websites. And that doesn't mean just clinicaltrials.gov, although that's what everybody does. You need to bear in mind that there are another 16 or so trial registries. So not everything is on clinicaltrials.gov. And the WHO has a platform, this ICTRP, which you can check, which gives you access to all of these trial registrational databases. So you can really figure out what other people are doing. And it's important you make sure nobody else is doing the same or a very similar trial. Or if they are, is the duplication really justified? If you're replicating an existing trial without strong justification, before you finally give up on it, it's worth checking that the other trial is actually happening, because sometimes people register trials before it's actually ready to start or before they've even secured funding, and sometimes they just never take part. So if you've got a great trial idea and you're about to give up on it because it looks like somebody else is doing it, maybe just worth contacting the investigator and make sure that it really is going ahead and what stage they're at. If it's being done already, then stop and move on to another idea. So that's about coming up with an idea and really honing down to the best trial question and making sure it's original. The next piece is, I'll just say a few words about designing a trial. So this is a critical thing, and people tend to want to rush it. Once you've got a good idea, you want to rush into starting your trial and getting funding for it. But really, you've got to plan a trial very carefully, because trials can be really important if they're done right, but they are very expensive to do. They're very hard to get off the ground, and they're very easy to go wrong once they have started. And then once they're off the ground, it's very hard to fix if they do go wrong. And if they go wrong, they can cause serious harm. So it's really the planning of a clinical trial is far more important than, say, a cohort study or any kind of laboratory research where it's much easier to fix as you go along. The trial, very hard to change once it's started. So you've got to get the planning right. There are lots of different aspects of a trial to continue to consider. including defining the intervention. It needs to be very precise so you're sure you can replicate what you're actually testing. You need to be able to define the patient population. Is it going to be very restricted, like in an efficacy trial, or the kind of thing that you might be doing is more an effectiveness trial? You're trying out an intervention in a more generalizable population, so you tend to need broader eligibility criteria, and you need to consider other factors shown here. Trials shouldn't be discriminatory, and avoid vulnerable populations. And then defining the outcome parameter, also very important. You can test lots of things, measure lots of things, but you have to define one of them as being the single most important thing that you're going to evaluate your intervention on, so a single primary endpoint. Key thing with all of these things is you need help in designing trial. If you're not experienced, and even an experienced investigator, trials are still better if you collaborate. Really, you have much more success in trials. The key is collaboration, and that starts with designing the trial. So it's not just implementation. So even if you don't want to work with some people in designing your trial, just make sure you do if they've got skills to bring or help to bring. And what kind of collaboration do you need in designing your trial? Well, the key is a statistician, and that's not just for sample size. Very often people think, oh, I just need a statistician to make sure that I've got the right sample size. But no, statistician can add much more value than that in ensuring that you're going to collect the right data in the right way to answer your trial questions. So do engage fully with a statistician for the whole trial process. You need local clinicians and epidemiologists to make sure that you have enough patients around and access to those patients to meet the eligibility criteria and meet feasibility requirements, and whether your safety and efficacy monitoring is appropriate. And community representatives, also very important to bring in early on, tell you whether the trial's worthwhile from their perspective, whether it's acceptable. And then a trials unit can help you with reviewing a draft protocol for feasibility and remind you of essential things that need to be in there to satisfy GCP. So all of these people and groups are very important to bring in at the trial design stage. So lastly, some pointers on obtaining funding for a trial. And here there are really three main options. So there's pharma funding, there's academic funding, and then a combination of different sources. So pharma funding, essentially at most times, if you're coming with the idea, you're talking about an investigator-initiated trial. And usually that's really only for licensed drugs, because pharmaceutical companies usually want to control their drugs pre-registration. So usually you can only get access once drugs are licensed. And the best time to secure support is soon after a drug has been registered, when pharma companies are usually looking to broaden the evidence base to enhance generalizability and looking at different populations and risk groups, and they've got resources to do that. So that's the best time to ask as a drug is coming up to registration or shortly afterwards. And most companies have a formal review process, so it's just a matter of approaching your local representative for drug X that you're interested in, and they may be able to give you valuable initial feedback to help shape your proposal to give it more chance of success when you formally submit it. And you can request support for drug only, which is relatively easy if the question's good, or if you want money on top, it starts to get a bit harder. But those are the options. Academic funding is difficult because trials are expensive. So local funding agencies may not have sufficient resources to fund the whole trial. And often if there's no separate funding stream for trials, you're mixed in with general funding stream, it can be quite difficult because one trial can wipe out all of the available funding for many laboratory research projects. So other researchers can get upset both with the funding board and with you if you end up taking all of the money. So that can be challenging. International funding agencies, of course, have the resources, but the competition is tough. And here, again, it's really important you have a strong collaborative team. That's often the deciding factor in success. So again, important to get your collaborators right. And then the last thing is a combination of many sources. So think about taking advantage of routine clinical services. So if you can do your trial within the routine HIV care services, that may reduce your costs substantially for routine safety tests and drugs. As long as the government provides that care free of charge, you can't expect patients to pay for their treatment to be taking part in a trial. You can consider deferring costs, for example, storing samples in anticipation of getting funding later on. But your trial must still be viable just in case you don't get that extra funding. You can consider borrowing or sharing manpower. You do need a trial manager or research nurse to help you oversee accumulating data quality. It's very hard to run a trial, even of modest size, on top of a busy routine clinical job. So you need somebody to help you with managing the data and overseeing the data collection and the running of the trial. And in reality, many trials use a combination of pharma, academic, routine clinical care, borrowed or deferred costs. There's no shame in that. Because they're difficult and expensive, we often cobble things together. But the key thing is, don't attempt a trial if you really don't have sufficient resources, because it won't be a happy experience and it puts patients at inconvenience or risk unnecessarily. So good luck with your efforts. I hope those pointers have been useful, and I'm happy to pick any of these up in questions later. Thank you.